So I'll just start out with a question. We've just heard a couple of speakers who talked about ways that they incorporate RNA information into their work. And there are a lot of ways, I think, that these two kinds of information prove valuable. And I wanted to just ask the speakers to talk about how you see, RNA data of different kinds, whether it be spatial imagery, single-cell RNA-seq data, or other types being useful in your work. I don't know if we want to start.
I feel like we should start with you.
Yeah. Hello? Oh, okay. So I think RNA information is very valuable because it gives us a preview or a picture of what is happening at the transcriptome level, because sometimes you have differences in expression of the transcript versus proteins. So we, in my work, for example, I didn't talk about that in this presentation, but we also study DLBCL, and in DLBCL, for example, there's a huge component of EBV is driven by Epstein-Barr virus, and for some types of DLBCL, and also in T-cell lymphomas. And by knowing about the RNA expression of these viral proteins and combining that with imaging of those proteins of those viruses in situ in those tissues, we get a better idea of what is happening within the tumor microenvironment. So it's very useful for us.
Mm-hmm. Santosh, we were just talking about this in a different setting. Do you want to pick that up?
Yeah. So I'm actually part of a single-cell biology lab. So we offer a lot of single nucleus RNA-seq, spatial transcriptomics, and also platforms like the Xenium, high-content FISH technology. So what we try to do with researchers who come to us is do single-cell or single nucleus RNA-seq, whatever fits your tissue type, get that data, and that forms a really important data set for informing panels downstream. You can then go ahead and profile a lot more samples using imaging, right? So RNA-seq data, I think, is a really important tool for helping with panel design.
Can you talk about that a little more? That seems like a pretty interesting area.
Yeah, so when investigators come to us like with tissue or disease tissue of interest, they want to study, a lot of the time, you would think that they would have a handle on exactly what 40 marker panel that they want to use to study the tissue, but that's actually not the case. A lot of time they were like, okay, this is the tissue setting that we want to profile. Like what can you do on your platform? So in that case, we start them off with a backbone panel of sorts and say, because we have seen multiple different tissue types, we'll say this is something common. These are the targets that you can look at, gives you a good overall view of your tissue and also maybe disease. It could be cancer or something else. And maybe that's a 25 marker panel, maybe it's a 20 marker panel. And then you can say, you have 15, 20 spots open for something of your interest. Are you interested in signaling? Are you interested in T-cell subtyping or dendritic cell subtypes? They say yes, but they don't know what exactly to look for. And that's, I think, where single-cell RNA-seq can really help. Mm-hmm. So you do your differential expression analysis, disease state, normal state, you find certain clusters of interest, and then you come with that to us, and then I'll help them with looking for specific markers for that cell type, validating those markers and incorporating that into panel design. So I think that's a way where RNA-seq can be useful. And there's also spatial transcriptomics via Visium, where the proteomic data via the phenocyclic fusion it can be really complementary to that analysis. Even though you get spatial data and RNA data with the transcriptomics, it's really not single cell level yet. And also, I think just having protein expression data is important because it sits above that regulation layer. So it's important to see protein in the end. So some people say, oh, I don't want to spend all this time with marker validation and running this. I'll just do Visium. But I think it's not an either or. They can really be complementary technologies. And the questions you can take from Visium, you can answer with your protein expression data at single cell level. And that can lead to further questions you can take downstream and find different angles of interrogation of your transcriptomic data. I think it can form a whole ecosystem that way.
Yeah. Do you find folks coming to you where you'll have issues of sample heterogeneity, or is that generally not so much a factor in your work as you're kind of trying to map these things out? I'm not sure what kind of samples are presented to you in your setting.
Yeah, so we do all types of different tumors. We do like pancreatic islets, people doing diabetes work. So yeah, it is challenging to optimize panels, and it's not just optimizing antibodies for target based, but it's really tissue dependent. Staining patterns differ, the workflows kind of differ, the autofluorescence level differs. So you have to take all that into account for when you're trying to kind of design and optimize panels that way. So some markers which are there in some panels may not work well. So we need to work with them to say, okay, you need to replace these and look for something else. In order to get good quality data.
Oh, hi. Yeah. So we work in our lab with protein-based analysis and also RNA-based analysis. I think, as he mentioned, I think RNA-based analysis are kind of complementary to the protein-based analysis, because as I'm going to show on my presentation today, these was the questions that were left open after we run the protein-based analysis in our assays, and then that we're able now to start thinking about mechanisms behind the findings that we had using RNA-based analysis. So I see them like complementary.
Can you talk a little bit about the techniques you intend to use to ferret that out?
Yeah, so for protein-based analysis, we're using the phenoimager, but for RNA-based analysis, we're using digital spatial profiling and now cosmics too. That has this single cell resolution that we don't have on geomics, for example, on digital spatial profiling.
And then once you kind of do that work, I'm interpolating here and looking for you to correct me, that will then feed back into the work that you do on the phenoimager in terms of your interpretation and your panels?
Yes, because as I mentioned, like the main, the questions that were left open after we started running this protein-based analysis, we now are able to start answering them and looking for mechanisms behind of that. So I see them pretty much complementary. So I'd say that we would start looking at the landscape using the protein-based assays, and then based on the questions that we have or the findings that we have, we then can move more targeted to the RNA space. And also important for also finding mechanisms of resistance, for example, which is very important in pancreatic cancer.
Understood.
As I see it, from our company, we don't care about big data. So the best data would be single cell RNA-seq. However, due to technical reasons in preparation, and also, let's see, single cell RNA, they have all this, let's see, economically, you cannot afford to have single cell sequencing every single time. So bulk sequencing is a way to go. To get accurate deconvolution, you have to use multiplex imaging as a major tool to help develop that. And right now we are working on it. And this will help you, let's see, in the future when you deal with, let's see, let's say, rare cancer types, and for which you have no prior reference. This is very important.
Indeed. Interesting. Any other thoughts or jointers?
Yeah, I'm happy to add just a little bit. Obviously, I didn't talk about any RNA work. Our group does do some, and there are some key modulators of the immune pathways that are not easily targetable by antibodies, and so they definitely lend themselves to RNA measurements. Our group, when we do it, we're not trying to do it at transcriptome level analysis, but rather doing just a kind of simple in situ hybridization. There's obviously lots of different platforms out there for that, but tying that together, obviously lots of groups are working to tie together at kind of even the lower numbers of targets level, the protein and RNA analysis. And it really certainly, there's clear cytokine factors and chemokine factors that we can't capture the protein information for, that will, I think, really inform some target validation in immuno-oncology.
Thanks. And I apologize for looking at my phone. It's where I've put my next question. So in the talk that I'll be giving later today, I'll describe how Akoya is focused on making spatial biology imaging more practical and faster and more flexible, so it can be used sort of in more routine and in larger range of purposes. And I'm curious as what other issues, beside the kind of, I'll say, practicality and speed issues, do you see as being limited? What issues, particularly perhaps beyond sample imaging, as being limiting in getting information out of the sample sets in your work?
I saw this question and I didn't
want to answer.
I didn't have an answer, but I'm holding the microphone. So I think, for us, obviously, what I presented to you is we're using the Akoya, the Opal Floor and PhenoImager system, which is limited by numbers. So for us, we still feel very constrained. And even though we can do 6, it's always, people always want more, of course. So I think for us, that's our biggest limitation. I guess scientifically, that's the biggest limitation. And then kind of implementation-wise, I think for us still the timing of analysis the timing that analysis takes is obviously still a struggle, which is kind of inherent, I think, in imaging analysis.
Yeah, you talked about, I'll say, a very image-centric, as opposed to some of the talks we heard were ones where AI or other tools do the segmentation, right. And I think that's what you're talking about.
Correct.
Okay, and you work with, as I recall, both sort of an informatics as well as a statistician group. Sure. Can you talk about would they say, they have a lot of questions from their side beyond the ones you've just mentioned?
I'm sure they do, especially the computational biologists, although I'm not, I guess I don't know well enough how they're thinking about it to be able to phrase them. But certainly the pipelines that they develop as we do approach being able to do more and more markers, the way they handle their data might have to look different, and so that's something that we'll be considering.
Yep. Archie, your thoughts?
I think the issue or limitation we have is like throughput, because we have a lot of samples to run, but Akoya already addressed that with the PhenoCycler-Fusion 2.0, which can run 2 slides at the same time. So in theory, we don't have that limitation if we buy it. So hopefully my PI would make that purchase soon. And after we generate, we run a lot of samples, and we sometimes have issues with like analyzing all of them, because some of the sections are really huge, so it takes a long time to segment them. Usually I can't even load them on my own laptop because they're pretty big. So I think those are some of the limitations we sometimes face.
And your work is all done on local PCs or on a local server or on the cloud? I'm just curious how you're set up.
So if it's a pretty small enough sample, I can do it on my PC using QPAD. But if it's a really huge sample, we run it on HPC for the segmentation. And there are also other publicly available cloud-based segmentation tools like DeepCell that we also use for our segmentation sometimes.
Yeah. Thanks.
Yeah, we're in a more like a research setting, so throughput is not really a big concern for us. Most of our time is just spent designing and optimizing panels. Someone will come with us with maybe 12 samples and say, I want to build this whole panel to profile these 12 samples. They put 2 samples on a slide, so there's just 6 slides for us to run. So I think the current system kind of works for that. The boring answer is cost for, as with for any research institution. Building out new panels is an expensive endeavor. So we kind of try to mitigate that by eating some of the costs ourselves and developing, like I said, backbone panels that are common across various tissue types. And then we can help researchers charge them out like a per test cost versus, okay, now you need to buy all the antibodies and all the conjugation kits and do everything. You profile 12 samples, and then your panel is just in the fridge for 6 months. But again, echoing the other points, analysis is a big issue. I think there are many very powerful open source tools available, but I think what is missing is a biologist-friendly, simple first or second pass tool that, you load up your QP-TIFF, it just gives you segmented phenotypes and you can click around and see, okay, these are neighborhoods here. A pathologist can take a look at it. They can see data that they can make sense of, get clues on what to look for further. And then you can obviously do all the big data analysis and unbiased clustering and all that stuff. But I think visual analysis tools are currently lacking.
Is that because probably pathologists are a big constituency for you, or just for everyone to help understand things?
No, they're actually not. We don't really work with that many pathologists. It's more like research labs. We do collaborate with them for, evaluating markers and expression and stuff like that. But we work with regular researchers who don't all have bioinformaticians as part of their group. We don't have a big bioinformatics personnel within our group either. So we try to give them cleaned up, maybe up to like segmented data, but after that, they're kind of shopping it around and say, oh, who can we collaborate with to analyze this data set? Whereas if they had tools which they can just do easy first or second pass analysis, they might be able to pick up, in the least, like which direction they want to focus more detailed analysis on. So I think that's kind of a space that's not really full.
Right, to help guide somebody through that search, which is so many dimensional.
Yeah.
Yeah, so I agree in the increasing the number of plaques, but now with the fusion, we will have more antibodies to include, because the panels that we developed and optimized work pretty nicely, but they are limited to the number of antibodies. So if you have specific questions, you then need to run multiple samples. And in my case, that I work with clinical trial samples, where usually we just have sometimes 2-3 slides, we need to kind of choose specific panels to run. So definitely in our case, having more capability to run a higher number of antibodies would be preferable. In addition, we'd like also to see better improvement on AI tools for example, cell segmentation. I don't think, for example, the informed software also, as many image analysis software don't do a very good work on cell segmentation. And also particularly using inform, it's difficult to run batch analysis, for example. We use in our lab a lot of batch analysis because we need to run multiple samples, multiple TMAs, and you cannot create one project per TMA, for example. Right. So we use a lot batch analysis. However, when you go to review the images, we cannot easily change the phenotypes if you see, for example, some cells that were wrongly phenotyped. So basically, we need to restart a new project, create a new algorithm. So I think we could have more flexibility changing these phenotypes after running batch analysis.
So both the segmentation, which is sort of tedious and repetitive because it's iterative, it sounds like, and then the phenotyping implication of that, and why can't I fix up?
Yes. Because when you see, for example, you train your algorithm very well, even trying to include different configurations of the same tumor, different tumor differentiation, more or less of the specific cell types to create a very good algorithm. But even with that, once you run batch analysis, you see some kind of errors, mistakes, and then you cannot do anything at this point. You need to go back and redo your algorithm or do a separate project for these specific cases.
So for us, the biggest issue is to add new markers to the panel. So the customers always want to pick outside of the menu. So what we, on our side, we see what's the best way to put it there. Because if we restrict it to the fusion antibody conjugate, it will take time, barcode, channel, everything, and also not every antibody works with the method. So I'm thinking of, since you introduced last year about a phenol signature code, that's essentially combine the two platforms. I would like to see more of it. So it will be more flexible. So whenever we want a new marker, we can use the other platform to add on to it.
Sure. Meaning more breadth of content in that particular.
Yes.
Yeah.
As I see it, the biggest advantage of this phenol imager is its amplification capability, which for a lot of markers actually is absolutely necessary. So if it combine both, it will solve a lot of problems.
We know the next question that I had, which they've all seen, is awfully similar, but let's see whether it provokes different responses. You know, as I'm the vice president of R&D at Akoya, and so it's my job to oversee the development teams and what they work on. And so imagine you were in my shoes and you had a freedom, which I don't have really, which is to tell everyone exactly what they would work on and that they would do it. What would you have them work on that would be most valuable to you?
Okay, since I hold the mic first. So first, I think Akoya could take the lead in pushing MixIF into clinics. I know there have been a MITRE study for many years across multiple institutions, but I think a fusion or in the future, the phenol signature code would be a better platform to push forward. So if I would go that first.
Yep.
Yeah.
I think in our case, or thinking about the clinical trial samples, I'd like to see a platform that combines multi-omics analysis, proteomics, and spatial transcriptomics. So then we could get a lot of information out of just one slide instead of running one specific protocol per slide.
Right.
Yeah, I can echo that. I think adding some RNA capabilities to the assays, that would be helpful. Ideally more than the 12 that RNAscope helped us to do.
You would use it for the kind of purposes you spoke of earlier, or for, okay.
No, for the same ones. Like Kathleen mentioned, there are many targets for which there are no good antibodies.
Sure.
Right? So, or they're like secreted molecules or cytokines or stuff like that. It's really important to have RNA, and that can really, a lot of times we've seen like that can make or break a panel or for that particular researcher's question that they want to ask. They have one or two of these which are like really important, and despite our best efforts, we can't really find good reliable antibodies. So RNA integration would be nice. I think the whole field kind of going that way. Every high content RNA profile is adding protein. Every protein profile is adding a bit of RNA. So I'm sure they'll all merge at some number in between. And in line with the cost issue that I mentioned, it would be good if Akoya had a bigger catalog of pre-conjugated antibodies for, there are like some very common targets that are absent in the catalog. So I think that would be a huge cost savings because we have to go custom route for all of those.
Right.
So if that is available, that can like half our cost and investment, and that it's not just about what we're paying, it's about what that cost means for the researchers and getting them to do or consider the assay. It's a make or break.
Right, because you're presenting them with spend this and we'll see instead of a more of a known quantity, and so that's a harder thing for them to weigh and evaluate.
You know, even if we can guarantee that it will work, even getting past the spend this is an issue. So if we had a easier entry way, that would be, I think, really useful, and also bigger, I think, mouse catalog. A lot of people are, at least for us, as part of the Jackson Labs, we're getting a lot of interest in archival mouse samples. We have huge projects which are hundreds of samples big, which they want to now include spatial transcriptomics and protein profiling as part of that consortium-like effort. So I think just improving the catalog of available antibodies, that would be good.
Yeah. Archie, what are your thoughts?
Just to, I guess, re-echo what the other panelists said, I think included RNA capability would be very helpful for us. Then this is just a pet peeve I have, but sometimes if an experiment fails, you have to start again from the beginning. I wish the software would allow you to even pause an experiment, because once you start going, there's no going back until it hopefully finishes successfully. So
what would you do if you paused? Why would you not want it to run to completion? I'm just curious.
So for example, sometimes you would encounter a failed experiment for an issue with like image registration or something that is not too, I would say not too critical.
Yeah.
But at that point, you would have to, if there's an error, you have to start again, and you have to remake your reporter plate with.
So time and the lost
Yeah.
setup. Yeah.
Yeah, and the cost of the reagent, which you have to remake. But I know it's a very technical issue because I've spoken to Akoya support and come back and forth. So I understand the technical challenges with that, because you have to make sure you can accurately register the images from each cycle. So I don't know what the solution to that would be, but it's just.
Sure.
Yeah.
Well, I asked you what you wanted.
Yeah, just that flexibility. I don't know.
Yeah, the flexibility of that. Okay, thanks.
So that's interesting, and I don't do codecs, but it sounds like you want it to kind of have some kind of built-in testing that you can tell if the experiment is working well. And if a problem comes up, it kind of alerts you, and you have the option to abort the rest of the staining or something like that. That's interesting. I can see how you'd want that. I can't think of anything beyond what people have described. I think obviously the incorporation of RNA is really useful. Our group does talk a lot about the ideas that were mentioned of kind of doing one standard assay with something like the phenocycler fusion, and then using the OPAL system after as a way to target maybe more sensitive antibodies or more targets that are unique to the project that are being asked for. So that I think I think you guys are working on it, but I think that has a lot of promise for the future. And then it has both the standards and then has flexibility also built into it.
Okay, so I'll go from maybe what were some very specific, narrow questions to the broader one, which is, where do you see these kinds of protein profiling, spatial transcriptomics kind of things having the most potential benefit or utility? Is it going to be in research? Is it going to be in guiding clinical tests development? Is it going to be in clinical test deployment? And so kind of in your space, where do you see the potential and where do you think we are, as we go from just starting to being there? Where are you in your particular part of that space?
Yeah, so I think my group does certainly some level of research, but then obviously is involved in clinical trials like we described. And I think I would like to think we're kind of midway on the path to having established clinical assays. I think there's a lot of merit to them in the field. I think I can tell that there's a fair amount of interest in them, but has been challenging to implement. So, I personally, obviously that's the work that we're doing and we're interested in, but imaging is amazing because there's so many other, areas of research that you can apply it to, so.
So we primarily use it for research, but I think it could have really beneficial clinical impact. For example, in my one of my projects on lymphoma, I don't know too much about the clinical aspect, but I know when a physician is making a diagnosis, they would have to do a immunohistochemistry for a bunch of different markers. So you do them on adjacent slides, and even the tissue section begins to look a bit different as you go through the blocks. So if this, like using the PhenoCycler could be part of the clinical workflow where the physician would be able to visualize all these different markers simultaneously and leverage that to inform their diagnosis or their clinical work. I think that would be very...
So building the reflex tests into the basic initial test suite. Yeah.
So that would be very useful.
Yeah, I want to echo what Archie said. I think it's potentially useful in the clinic, but not to be too pessimistic, but I think we're a ways off. I think you'll have to pry away the histologists from their LICA bonds before you can get anywhere along that path. But yeah, it all comes down to reproducibility and also how well can you interpret the result, right? That's why we're still doing single marker IHCs. It's easy for one person to sit with a computer screen and score or look at a score and decide. I think any advancement will include a heavy dose of machine learning, AI kind of magic. We can definitely get technologies there where you can do 60 markers, it gives you an image, but then how are we going to analyze it? Like the Boston Gene folks were saying, like what do you report out? What are the key metrics? How do you use that information? I think that is the biggest bottleneck.
Sure. And so you can get the insights, but you can't get the binary 1, 0, exactly. 1 + 2 + answers. Yes.
Yeah, and we've worked with multiple clinicians on different research projects, and if they can't get that answer, they don't want to do the study, right? That is they'll view everything through that lens of, okay, what can I take from this to the customer, to the patient, and how it's going to influence their daily decision workflow about patient stratification, treatment decisions, etc. So if we can't plug into that, it doesn't matter how much data we're generating, how many pages of reports we're making. It kind of needs to help them with one of those clinical attributes that they're looking to advance. I think the technologies are there. It may not be this exact version of the PhenoCycler Fusion. It needs to be much simpler, much easier. Somebody can just load a slide in, stain it with the lyophilize, pre-made cocktail, labeled breast cancer screening panel 1, and get the results, feed it into an AI model, which shows them the numbers that they're used to seeing, and then helps to stratify that way. Until we get there, I think it's still quite a ways to go.
Yep.
Yeah, in our case, we use mostly the research space, but also the results that you get using these technologies were also used to guide openings of new clinical trials. So I think this was like coming from the information that we're able to get out of these technologies.
To guide trials and those kind of,
Yes,
yeah.
yes.
So for us, you can see we are pushing for clinical use of this. Just this is not enough. I think it's a two-front. For research and for clinical use, they have different standards. For clinical, we realize it's, let's say, as necessary as possible. You don't need a huge panel. For research, is as many as possible. So, so we're facing two fronts, and we have to deal with collectively. Yeah.
Good. Those are the questions, and I think we're at the hour. So I want to thank you all for your, boy, this is really boomy. I want to thank you all for your observations and for your insights. Thanks.
Thank you.
Thank you.